Hello and welcome to Cambridge, Massachusetts. Tonight I have a very special guest. Marc Vidal is the director for the Center for Cancer Systems Biology at the Dana-Farber Cancer Institute, and is also a professor of genetics at Harvard Medical School. And Marc is a very interesting guest. You know, he's one of the first persons that I met when I actually came to Boston. And he has been working for many years in something that is very interesting, and it's in the field of genetics. Most people is familiar with this idea of the Human Genome Project, this effort to map out every gene, of humans. And there have been projects also to map out genes of many other organisms. But Marc has been thinking about how these pieces go together, has been building this jigsaw puzzle. And he heads the Human Interactome Project. And I would like Marc to tell us a little bit, what's the Human Interactome Project about?
Okay, well, first of all, thank you for having me, Cesar. It's very nice of you. It's a very nice setting. So the Human Interactome Project, and by the way, as you just said yourself, the Human Genome Project was extended to other species as well, and we'll go back to that, I'm sure, during this interview. But the Human Interactome Project is about putting the pieces back together. You're absolutely right. So basically, how does this work? So when you hear on the radio, watching TV, or reading a newspaper that a particular gene was discovered that has a role in breast cancer, for example, what exactly does that mean, right? So the first big question that most people who are not geneticists, who are not scientists, actually are asking themselves is, why is it that people have bad genes? I mean, as a matter of fact, if we believe the theory of evolution, we would expect that actually at this point of our history of the humankind, we shouldn't have, quote, bad genes anymore. It turns out there really are not any bad genes to speak of in our genome. Instead, what it is that genes make products. They're called usually proteins. They could be called RNAs as well, depending on the cases. The Human Genome Project, as you mentioned, identified about 20,000 protein-coding genes. And it's true that when we say this gene is involved in breast cancer, it has a relationship. The genotype-phenotype relationship is meaningful in the sense that a mutation, a change, a deficiency in a particular gene, which, by the way, otherwise is a good thing for you, right, might give rise effectively to a high susceptibility to a disease. Now, what people realized a long time ago, this is a few decades ago, people realized when you think about it, genes or their products, proteins or RNAs, cannot really do that much alone. It doesn't make any sense, right? How could a molecule by itself, whether being a gene or a protein or an RNA, how could it do anything in the context of a cell, a very complex system? We'll go back to the notion of system in a little bit. How could a gene do anything by itself? So a bunch of people, including myself, I would say almost 2 decades ago, started thinking, well, if we are really in the process of finding all genes, we as a community of scientists, if we're really in the process of finding all genes, again, we know now that the human genome has about 20,000 protein-coding genes, wouldn't it be interesting to try to figure out the wiring diagram of how all these genes and their gene products interact with each other? And this is the key word. Like I said, we don't know of any genes or proteins or enzymes or RNA that can do anything on its own. So that presupposes, obviously, that all of them have to interact with a certain number of other genes, proteins, RNAs, enzymes, etc., to do anything. And at the end, when you start thinking of how this could actually be drawn, even just simply representing this reality, it's mind-boggling, right? So you go, what is the wiring diagram of the cell? Could there be a map of interactions that would explain, that would underlie the behavior of a cell, the behavior of an organ, the behavior of an individual? And so that was the basic question 20 years ago, is there going to be a way for us to basically build, sometimes people say stand on some giants' shoulders. In this case, the giants are the people who map the human genome. And the information that they generated, could we stand on these people's shoulders and on the information they generate to come up with the next level of complexity, so to speak, going from a parts list, which is in a sense what the 20,000 genes represent, going from a parts list to a wiring diagram.
And in these last 20 years, how much progress have we made in trying to map out these wiring diagrams? You know?
That's a very good question. And I started doing this when I was a postdoc in 1993, so it's almost 20 years ago. And I think if I would have known then that it would take us so long to get to where we are, I'm not sure I would have dared actually doing it. But at least I was young, and and I just didn't realize how complex things were going to be. So to answer your question, we've measured this. So the first thing you would need to have to answer this question is you would need to come up with a denominator, so to speak, which would be how big is the interactome, right? So let's redefine it just to make sure, right? We went through this after your first question. Basically, for us, the interactome, the human interactome network, right, is the complete network of all macromolecular interactions in the cell that is mappable using the information from the human genome sequence. We've measured with a bunch of colleagues, including Laszlo Barabáshi's lab and others, we've measured that the approximate number of protein-protein interactions in the human, encoded by the human genome, so to speak, in the human interactome. And we think that number is about 130, 140,000. So let's summarize. The idea is we have 20,000 protein coding genes. We think there must be about 130, 140,000 interactions mediated between them. For those who know really the details of genetics and biochemistry, those numbers do not include splicing isoforms. That's a level of complexity that we're not able to measure quite yet.
And how much would the complexity go up if you start including all those variants?
Very nice. So we actually have probed this a little bit at the level just simply of the genes, right? Again, so the components, the parts list, and others have done that very similar types of work. And we think there might be between 5 and 10 isoforms per gene. So that would mean that out of the 20,000 genes of the genome, we might get 100,000 to 200,000 proteins out of that information already, right? So increasing already the complexity by simply making the list of the parts longer than we originally thought it would be. And then how does that play at the level of the number of interactions? That's not really clear at this point quite yet, although we've started to measure this. I would just a wild guess, okay? Probably another factor of 5 after that, right? So basically now instead of having 150,000, let's say, more or less, you multiply that by 5, given the number of isoforms, you're approaching somewhere between 500,000, maybe 1 million protein-protein interactions. Now, what's interesting, you're in the field of networks, is that to biologists, certainly from the biologists that I was hanging out with 20 years ago, that's a huge number. It appears to be like such an incredible number. Nobody's going to be able to, one, map these things. We'll get back to how we actually do that. And also, if we can actually map those interactions, again, 20 years ago, people were saying, well, what are you going to do with the information? Remember, we didn't have the informatics that we have today. The internet was very, very early, and people weren't really thinking in terms of networks, etc. And so, at this stage now, we imagine that this number is somewhere, let's say, between 200,000 and 1 million. Where are we? That's the answer to your question. Finally, that's the denominator, right? Well, we have probably between what's been published by individual labs in the literature and the sort of efforts, sort of high throughput efforts that we're running, we probably have 10, 15% of the network, right? So somewhere between, again, the network without counting the isoform. So we probably have, between 10 and 20,000 really good quality, high quality protein-protein interactions. So again, from a networks perspective, now again, the biologists might actually say, well, that's very little. How can you do anything with 5 or 10% of something? But as you know, for many networks that are being studied by physicists or mathematicians out there or engineers, I mean, having already 5 or 10% of, as a sample of the beast that you're trying to characterize is actually already useful. And so that's where we are.
And then I think we can move to a variety of questions, but before we start, I think it would be useful to understand when you talk about an interaction, what is an interaction, and then how do you measure the interaction, like in the lab, what goes on, how can you eventually say, you know, gene A and gene B are working together?
So it turns out I was at a party last Saturday, a party for a really good friend's coming out of a book. She just published a book. In any case, there was an individual there at that party who was telling me that he's writing a book. He's writing a book on Lego, the company Lego.
Nice.
And so we had a really long conversation about that, because that's the analogy is almost perfect, right? So when you think about it, right, one way that you can build, let's say you're a child, 5-year-old child or a 10-year-old child or 40-year-old child, whatever. Let's say you want to build a castle, right? So one way that you could build a castle and play with it is you could have one part, right? I mean, you could imagine a way, boom, one piece. But there's no fun, there's no dynamic possible.
You cannot adapt.
You cannot adapt. Exactly, I was going there. Evolution, right? And also, you cannot replicate, right? It would be hard to make two, let's say. The analogy is not perfect. But once you reduce it down to smaller components, right? Now you can start playing, you can change the overall shape, you can adapt, that's right. You can have something a little bit bigger if you need to, etc. But then the question is, how do you deal with those components? And so again, one way that you could do it is do like sort of, bricks that would be attached to each other with mortar and, and cement. And then again, so then you can never go back. There's just, you have one direction, you build a castle, and that's about it. While Lego is really beautiful because you can build it, and as you know, you can unbuild it, you can destroy it, you can change it, you can adapt it, you can do all these things. And the beauty of the design, and the guy was actually telling me about, how the work that's going into the engineering of how the blocks are fitting with each other, the little bricks, right? It's really not trivial at all. And so I think it's a good analogy for those who don't think of genetics or biochemistry every day. Proteins are basically Lego blocks. And they like to interact with each other. They do so relatively quickly. Under some conditions, they can stick to each other relatively tightly, but it's also relatively easy to pull them apart. You don't need to change very much in the conditions of the life of a protein to change them apart. So it's an ideal way that nature came up with, evolution came up with, I think, to be able to build structures, respond to the needs of evolution over a long period of time, but also adapt to different environments, right? During shorter periods of time, temperature changes, whatever, right? So you have to readapt, and the cells have to react to that. François Jacob, he's one of the most famous biologists, actually a systems biologist in his own sort. In the 50s and 60s, he won a Nobel Prize. He always said that nature tinkers, right? That evolution is based on tinkering. And that tinkering is really ideal and can take place ideally if you have parts or blocks, Lego bricks, that can relatively, interact with each other in relatively precise, tight, and yet dynamically regulatable ways. And so that's what protein-protein interactions are all about. Now you were asking how you measure them?
Exactly.
So so you have two ways to do it, basically, and I'm sure we'll go there in more details. But overall, you can do it outside of the cells that you're studying, or you can do it right there, in vivo, inside of cells. So the idea is, can you find techniques that will tell you how two macromolecules actually touch each other right there in the cell as they are functioning, and the two strategies are complementary to each other. Or could you imagine a way to actually take these two proteins out of the cell, or actually, even better, make them yourself? So imagine you're a Lego engineer, and this basically what you receive is the blueprint for these parts. And so for the people who really know the details of this is the Orpheum project, basically. It's a huge effort that we started, and others as well, 10, 15 years ago, which consists in cloning every single one of the human genes. So sequencing them and finding them, the Human Genome Project, as you started with, is one thing, but then it turns out that it's actually not trivial at all to extract the genes out of the genome and make them into a form that they can be used to make the corresponding protein.
So now you have them in the shelf.
And now it's actually shelved, exactly. It really, as a matter of fact, not only that, I'll tell you, this is really going to the practical stuff, but I think it's interesting. 10 years ago, we realized that in order to organize all this, we needed to go to barcodes. You go to the store, click, you have the little barcodes when you pay at the cashier. And that, of course, the beauty of that is that the cashier doesn't have to type the number, but it also, as you know, is great because all the organization of the stores, you keep the stock, you can do statistics, you can take all the supermarkets of a country and you know about this. This is part of it. So we have all this now. We're basically equipped to do this at that level. Yeah.
So now that you have all the genes in these shelves and you can have a team of people take them out, how do you actually do you put them where to see where they stick or who sticks with whom?
Right. So basically, what happens there is we actually use a trick. And the trick is the following. And the trick was invented by a person called Stanfields in the late 1980s, 1989 to be precise. And the trick is the following. It turns out, remember I was describing proteins and how they like to interact with each other in ways that are highly regulatable, etc. Well, one more property that proteins have is that they're modular. In other words, many of them, not all of them, but many of them, you can cut them in two pieces. And both pieces will actually find the right shape. And if you basically, Stanfield's reasoned, if you were to bring them back together, one way or the other, they would actually reform a functioning entity. They would reform a functioning protein. So it turns out the basic idea is this. You take a protein of interest, you cut it in two. Now, neither fragment can actually work. And so what you do then is you fuse to the first fragment one protein of interest, you fuse to the other fragment another protein of interest, and if protein 1 and protein 2 like to interact with each other, they will tend to reform the function of the tool protein, so to speak, that you decided to use at the first place. So usually that protein is a transcription factor. We're getting into the details. But the point is that you can actually do all these manipulations over tens of thousands of genes, no problem, if you use a very easily manipulatable organism. And the choice is yeast. So yeast cells, the yeast that we use to make bread or wine or beer, are amazing organisms that we as a community of scientists have learned to use for the last half century or something like that. And so what you do is, to summarize, you trick the yeast cells, you introduce human genes inside those cells, and if the two human genes encode interacting proteins, the yeast cells will tell you that. It's basically assays that we can run very quickly on the yeast cells, and the yeast cells will tell you, well, you gave me these two proteins, I'm telling you that they can interact, or not. Okay. And so since it's, again, relatively easy to use yeast as a experimental organism, we can run huge numbers, right, going through the assay.
And then now that you are able to start creating these maps and you're able to start looking at the maps for different cells in the body or for different organisms, what are the difference when you have a different cell type or when you have a different organism? Is it more or less a change in a new Lego piece, or is it a change of the way in which these Lego pieces are connected? How is evolution dealing with this trade-off?
Yeah, so that's a really good question. I think we could answer to be relatively simple here, to simplify things, that is both, I would say. So one way of regulating things and creating cells of different types would be to regulate the genes that are expressed in those cells, the genes that are being made in each one of these cells. So let's say with the same genome of 20,000 genes, you could differentiate one way and become cell A, or differentiate this other way and become cell B. And the way you become A or B is that a subset of the 20,000 genes are turned on in A, and another subset of the 20,000 genes are turned on in B. And so that happens, definitely, that's very clear. But of course, then what happens is the result of having the A subset of genes expressed or the B subset of genes expressed is such that that also has a direct effect on the network that takes place in A or in B. And by the way, that's sort of downstream of differentiation, but upstream of differentiation, it becomes more and more clear now that basically it's also the interactome network that dictates what subset of genes is going to be expressed in A or in B. So it's really sort of, as always in biology, everything is in a loop, right? So you have the genome, the interactome network, including the enzymes that replicate the DNA of the genome, the enzymes that transcribe the genes of the genome to turn on the genes. They are under a very tight regulation from the network, and at the same time, they induce different kinds of networks in the different cells as they differentiate, if that makes any sense.
And now when we step a little bit aside from the fundamental biology and go a little bit more into like medicine and that aspect, like when you start fishing with a net instead of fishing with a fishing pole, and you look at these networks of protein interactions, what do you find? Are there some insights that would be worth highlighting, for example, when it relates to cancer or other diseases?
Well, the first answer to that question is really that things are much more connected than we ever thought of before. I mean, until, I would say half a dozen years or so ago, I would say that people really thought of genetics, as I said at the very beginning, as a series of genes, and each gene could explain part of a disease or part of a phenotype. But it turns out things are much more complex. So genes are connected to each other through the kind of diseases that they are associated with. As a matter of fact, we collaborated with Laszlo Barabarshi's lab to come up with a really interesting map of diseases. It turns out that even diseases are connected with each other in very complex ways. And in that map, we call it the diseasome map. In that map, diseases are connected to each other if you know of at least one gene that's associated with both of them. And so what the lesson that comes out of this is that, on the one hand, the system that we're trying to study, cells, tissues, organs, individuals, and actually even at higher scales, is much more complex than we ever thought before. Genes, to summarize, are involved in very complex wiring diagram of interacting parts. And in order to understand, that's when everything goes right in unaffected individuals. And it's becoming clear that in order to fully understand disease, and I would suggest, although there is no demonstration of that, and I'm also not the only one to suggest that, I would suggest to find new therapeutic strategies to try to treat or cure diseases. It is increasingly necessary that we understand biology from a network's point of view. Again, because there's really no gene that's acting alone. They are acting in a very complex dance of macromolecular interactions. And so we see that in the diseases that have been characterized in the last century or so.
So if you look at any molecular biology book, like a basic book, the paradigm is kind of like, one gene, one protein, one function.
Yes.
That's sort of like
it's called the central dogma.
Exactly, the central dogma.
Yeah.
But now you're telling us that actually, the reality is far from that central dogma, that, one gene, obviously has variants, and that causes, it to be the predecessor of many proteins.
Yes.
But at the same time, those proteins interact in so many ways that they could participate in a variety of functions.
Yes.
So now, one question that I wanted to ask that it just kind of like came up is this idea that when you have Western medicine, is usually based on kind of like hitting one protein here or there, or like, it's like has one active component and it hits very, very targetedly. But you say there is a network, so in some sense, instead of like hitting it very hard here, would it make sense to hit it more soft in many different spots? And is this a little bit of like the different therapeutics that you talk about, or?
And are you maybe even thinking of alternative ways of thinking about medicine, such as, Chinese medicine or something like that? Perhaps, I don't know. So I'm not going to go there because I don't know anything about it. But it's interesting to think about the problem and speculate. So it turns out, again, it's sort of the glass is half full and half empty, I would say, right? As a, geneticist, molecular biologist of the 80s, I would argue equally that on one hand, the idea of finding genes responsible for disease and pathways, enzymes, etc., has been relatively successful in leading the way to discovering new compounds that, as you just said, act in a very reductionist way, meaning one compound is affecting one enzyme, and then we expect a solution, we expect the problem to be solved. And there are amazing successes in this, right? I mean, you know, we know so much about so many things now, so many diseases, the molecular mechanism of what's happening, that it would be silly to say that the entire thing is a failure. However, having said that, and we as a community of scientists and doctors, etc., have already saved millions of lives using those reasonings. At the same time, and that's where the glass is half empty, I would say, is we still have amazing challenges ahead of us. Cancer is one example, right? So there are beautiful examples of great successes in cancer. But for every case of a great success, a discovery of, in one particular form of cancer, discovery of a compound and how to use it, for every example like that, you have still 10 cases where people suffer, and there is not much you can do, at least in the mid and long term, to help them. We have, you know, protocols to try to extend their lifespan, and it's fantastic that we have them, but we don't cure many of the cancers. That's one example. Anyway, and then you can think of all the other problems for which there is very little done, like autism, complex diseases like that. So in other words, okay, so what can we do about that? Well, that's exactly the idea, is that there are traits, there are phenotypes, there are diseases that are relatively easy to deal with. You know, high cholesterol, right? Well, you know, you can take a statin, and we sort of understand that pathway, and that's one of the successes. Like I just said, cancer or autism, really a harder problem. And it's a harder problem because the function that those diseases are affected in are themselves very complex. And when you think about it, you know, we go from a single cell at the time of fertilization, right? An egg and a sperm cell. We go to a human body, you know, 10 to the 12, 10 to the 13 cells. So you have an incredible number of divisions, right, in a relatively short amount of time. And then at some point, you got to block this. You got to say, okay, enough, or at least go down, go slowly. You know, the brain says basically shut off. The skin says keep going, but at a certain rate. And so the mechanisms that are in place to, on the one hand, allow cell divisions, but also control cell division in ways that cancer does not occur, you can imagine them being extremely complex. So it's no surprise that we need to understand that complexity from the point of view of the wiring diagram. At least that's one important angle in order to go there. Now, does that mean that at some point we're going to try to find compounds that touch, as you said, more subtly different zones of the network, different types of nodes? Do we need to go after hubs, the highly connected proteins of the interactome networks? Should we instead go to nodes that are not highly connected? Those are still questions that are out there and are, as far as I know, unresolved. But they're fascinating, and I think the next decade or so will be really incredible from that point of view.
And I have another, I think, relatively simple question, and I just want sort of like a guess. I'm not asking for like a precise number, but it depends how you count, I'm sure. But how many cancers are there?
Oh boy, you mean how many cancer types?
Exactly, because you can have breast cancer, and there are many genes that are associated. So not
Sure.
all breast cancers are the same.
Sure.
Are we talking 1,000 cancers?
So first of all, full disclosure, I'm not an MD. I don't see patients. I happen to work at the Dana-Farber Cancer Institute, but I don't see patients, and I don't know the details of the problem. But I don't know so probably in the hundreds, I would imagine. And the reason why I say this is because we have about 200, people say there are about 200 basic cell types in the body. There are many more than 200 different kinds of cells, but if you, just the basic bio 101, skin cells, eye cells, exactly. So that's in the range of a few hundred. So I think many of them, if not all, I don't know the answer to that question, many of them can become cancerous depending on the environment and the genetics that a particular individual has inherited. So I would say, yeah, it's in the range of a few hundred. So in other words, maybe more important, because that we're all sure about, it's certainly not one disease. So when people say, "Oh, cancer, is cancer resolved?" Well, actually, we need many dozens of solutions because the problem is much more complicated than it's not a single disease.
Exactly. So just that we name it one disease is an oversimplification of a much more complex.
That's right. As a matter of fact, as you know, in reality, when you go to a cancer clinic, you have specialists of these different cancers. I mean, it can be different, many different years of training to be a good breast cancer oncologist relative to another type of cancer. So it's really a different problem every time you see a different kind of cancer. Yeah.
And now you've been in this field for many years. You said you started looking at interactome like around 1993, and
yes.
How has been the reception of systems biologists by like the mainstream biologist community? How has that changed over time? Yeah, I would like to get a little bit of your feeling of that, like someone that has been in the trenches.
Yeah, it was really the trenches, actually. You're right. So the short answer is that I think the last 15 years saw amazing changes in the way this is perceived. To be a little bit more detailed on this, so first of all, perhaps we should repeat one thing that I touched really briefly upon, and that is that there were really a bunch of genius people in the late 40s and in the 1950s and early 1960s who really defined many of the problems of what we now today call systems biology. There weren't that many relative to the number of biologists or geneticists or biochemists who worked at that time. There were really only a few, at least that we remember today. And so they were very smart people, and they really, I'm thinking, for those who know the field a little bit, I'm thinking Waddington. I mentioned François Jacob actually wrote a couple of pieces that are really interesting, Max Delbruck. And they realized that all the enzymology that was going on around them, meaning characterization of single purified enzymes, and try to understand life from that perspective, or individual genes, the central dogma, right? One gene, one protein, one function. That at the end, this was all very interesting, but it could never explain how a cell works or doesn't work. And the reason was, and they really had a bunch of theoretical justification for that statement, which I think were really quite amazing, actually. Starting from, small scale, how do you explain, how do you explain bistability? How do you explain that with the same genome you can have a differentiation between one cell type to another one, right? So Max Delbruck sits down and he draws a little network, a little motif, where he basically defines a positive feedback loop, for example, completely theoretically, right? And also, I'll skip the details, but the point is that in the late 40s, 50s, and early 60s, there were absolutely incredibly genius people who were starting to think about the problem. Now, what happened is, what they didn't have was the information that we have today. And you started this by mentioning the human genome. And really, for us, this renaissance, if you want, of systems biology was, I think, really the major factor that started that renaissance was the Human Genome Project. Because once you had 20,000 genes, once you basically had defined the parts, once you had the parts list, you could start dreaming. Now, it was still a dream in 1993. It was still crazy to imagine, for example, mapping all interactions between all proteins encoded by a genome. Just maybe one number that we didn't cite yet is the number of combinations that you have to test, right? So if you have 20,000 proteins, again, we're not going to talk about isoforms or splicing or anything like that. Let's keep things simple. With 20,000 proteins, you need to test 400 million combinations. You could argue 200 million, depending on how you want to do it. But if you want to test in both orientation of the...
So when you say 100,000 as the denominator is in a space that is mostly empty.
Exactly, right? And you have to find these very rare events in 200 to 400 million tests, so to speak.
So it's 100,000 needle in a haystack, bro.
Exactly. It's totally needles in a haystack, absolutely. Of course, the human genome sequence was not the only factor that helped restarting the idea of systems biology. I would say that the development of bioinformatics, the fact that computer science people started to come to biology and started being able to actually handle voluminous data, if you want, huge data sets. And then, of course, you had the physicists and the engineers and the mathematicians also who came along and started putting models together of different kinds. And there's still, arguments as to what the best way is to model cells, at the scale of a cell, at the scale of two proteins. We're talking differential equations, we're talking Boolean network modeling, we're talking about all kinds of graph theory ways that we can do it. I think at the end, it doesn't really matter because we're all basically trying to solve the same problem, which is how do molecules interact with each other to generate the states that the cells can have, to generate the phenotypes that they show. That's the summary of things. To loop back and finally answer your question, I think, many people in the mid-90s didn't always appreciate where this was going, I would say, at least in the biology community. Again, the fact that the genome sequence was released in the early 2000s, people started to say, well, okay, if we have that, I'm starting to see the idea. But as always, it's not a big surprise, and I'm not criticizing anyone here. I think everyone is very busy at trying to resolve the set of questions that they have in their mind, and that's fantastic. And if you have a new field that's sort of budding out, and well, that field has to demonstrate a few successes. And it's only recently, I have to admit, that the field of at least network biology, and in a sort of wider way of talking about systems biology, I think we're starting to solve fundamental questions of biology. And so now all biologists are interested in at least looking at what it is that we're doing, because they start seeing that there is really a potential future for this, that we are actually indeed solving fundamental questions. And if I may, I wouldn't mind closing this particular answer by citing an interesting set of reviews by another Nobel Prize winner. We cited François Jacob earlier. I have in mind Paul Nurse. So Paul Nurse won the Nobel Prize for his discoveries in cell cycle. You could argue, discoveries that were made one gene at a time, proteins, how they interact with each other. Just a couple of beautiful work, essentially explaining how basically cells regulate their cell cycle, talking about cancer, where that regulation is usually affected in the most terrible ways. So Paul Nurse has a couple of articles that he published in the last few years where he argues the following, and I really like that argument. He says, basically, and I'm paraphrasing here, I don't remember the exact words that he's using, but the idea is the following, is biologists accept at least 4 great ideas. The first one is basically the fact that life is based on chemistry. So it's hard to conceptualize the fact that there were biologists sometimes, a couple of centuries ago, who didn't know that life requires chemistry to occur, right? So they didn't know that, simply. The other great idea of biology, of course, is evolution, right? That's, as you know, 150 years ago, and natural selection, etc. So the third one is really the idea of genes, and how in order to have life somewhere, you require to be able to store the information. You said adapt when I was talking about Legos, remember? You have to evolve, you have to adapt, and for that you have to have all the information of your components stored somewhere. So that's, the double helix, DNA, right, etc. And so basically, so what's happening is that one thing that we actually didn't really conceptualize very well so far is that life also requires systems. That genes are not enough, DNA is not enough, even evolution by itself, if you don't consider the level of systems interactions between those the components of the cell, you really are not going to be able to understand the whole of evolution. So Paul Nurse, to go back to his argument, sees that as a 5th great idea of biology, which is only emerging now and is starting to become accepted by more and more people. So to finally answer your question, I think we've gone a long way in the last 15, 20 years. Again, it was not, the ideas were not completely new, They started in the 50s and the 60s. They were sort of left aside because people were lacking the data. They were lacking the data sets. They were lacking the tools to actually ask these questions. The human genome came in. Informatics started coming in. As you know, the late 90s, we experienced an amazing number of fantastic discoveries in the field of networks. So all this together now, I think we're starting to do something. We're starting to solve questions. And I think people are starting to realize that this might actually be, the fifth greatest ideas of biology, is that it's a requirement for life. I think that's the summary. Genes are required for life. Systems are also required for life. Networks of interactions are required for life. And if that's true, it is a great idea, right?
Exactly. So now let's go a little bit, to get to know Marc better.
Okay.
We learned about the way that Marc thinks and about biology, and now I would like to get to know Marc a little bit better. And if you could tell us a little bit about your story, like where did you grow up? Let's start.
Okay.
That far behind.
I grew up in the French-speaking part of Belgium. So as you probably know, we have two communities living together in Belgium. One speaks Dutch, or Flemish, and one speaks French. And so I'm from the French-speaking side of Belgium. I was born in Brussels, in the capital, and then my parents moved very close to Brussels. Let's say half an hour south of Brussels. And so it turns out that I grew up in the countryside, and I started really becoming passionate about agriculture, farms, tractors, growing things, animals. And so my parents didn't have a farm themselves, or neighbors did, and so I spent a lot of time during my the most difficult years of your life, the adolescence, sort of concentrating on working with farmers in the neighborhood. And so I really started liking this. And to make a living out of it, you needed to study that overall field. And so we have a field called agronomy in Belgium. It's not a word that you hear so much in English, but it's basically an ag school if you wanted to talk about it.
We also have
agronomy,
yeah.
right?
They put engineer at the end.
Exactly.
Engineer
well,
at
so
the end
same here. And now, by the way, it's been renamed bioengineering. But
okay.
But you know exactly. So basically, the point of it is to try to understand life from an engineering point of view and manipulate it and produce things out of it. And so from that, and my goal was to actually go to Africa. I was that was since I was a boy really. I wanted to go to Africa and basically help people, growing things and stuff like that. And then, and I'm sure you hear this a lot if you interview people like that. One key professor changed my life. Many people say that, right? His name is Arsène Burny, and he is a great scientist. He had a lab in that small college where I was doing my studies. And I essentially followed his class. I didn't really know what molecular biology was. It was sort of weird. And then I discovered DNA, double helix, genetics, genes. I fell in love with this stuff. And from that, I started working in a lab one summer in 1984. Actually, a yeast genetics lab. Remember, that's sort of the connection with the fact that we use yeast to map protein-protein interactions. And then little by little, I guess it's, essentially, that was the key. That's the interesting part of the answer to your question is one professor completely changed my life. And once I knew that I was completely in love with this field, then I was looking for the best place to actually, perform work in that field. And very quickly I realized that, the US was the place to go to. As a matter of fact, as I was walking through here to your place, I just remember that I was here in 1986 for the first time, and how things have changed and how it's amazing to be actually in this building 25 years later. So I did graduate school in the US, and then I came to do a postdoc at MGH, which is right across the river. And during that postdoc, it was with a person called Ed Harlow. I realized the importance of protein-protein interactions. He had made an amazing discovery. He had discovered that, talking again about cancer and cell cycle and regulation of cell cycle, he had discovered that the product of an oncogene, usually oncogenes force the cells to divide, and the product of a tumor suppressor gene. Usually tumor suppressor gene products basically are the brakes throughout cell cycle. They prevent cells from dividing. He discovered that the product of an oncogene, the protein made by an oncogene, and the protein made by a tumor suppressor gene could physically interact with each other. And I went to his lab to try to learn how to do that. And so that's where the idea came from. So I was there in the early 90s, and in the early 90s you kept hearing about the Human Genome Sequencing Project. This is very early on, right? They had 1% at that point maybe of the human genome, maybe less actually. But you heard it all the time that this was being done. And so I started thinking, well, the day we have the human genome sequence information available and we have the parts list, we've said this word a few times, now that I realize how important protein-protein interactions are in this particular lab where I'm training as a postdoc, wouldn't it be amazing to push the idea of interactions to the same scale than what geneticists are doing right now with the genome and try to identify all genes? So identify all genes is the Genome Project, identify all interactions is the Interactome Project. That's sort of how it started. And then I stayed in Boston, and here we are.
Very nice. Thank you very much for sharing all those wonderful ideas and thoughts with us. It was a pleasure having you on the show. Thank you.
Thank you very much.